Before this we have learned about the examples of some monosaccharides. As learning for today, we will discover a few things about the disaccharides and simple briefing about the glycosidic bond. Disaccharides are sugars made up of two monosaccharide units joined together by a glycosidic bond. A glycosidic bond is a form of covalent connection that connects a carbohydrate molecule to another molecule, which could be either another type of molecule like a protein or lipid, or it could be a different type of molecule entirely. Glycosidic bonds, as they are known in the context of carbohydrates, are specifically the bonds that unite two monosaccharide units to create a bigger carbohydrate molecule, such as a disaccharide, oligosaccharide, or polysaccharide. Dehydration synthesis, also known as condensation, is the process of removing a water molecule from the hydroxyl groups of the two monosaccharides being linked in order to form a glycosidic bond. Depending on the precise arrangement of the anomeric carbon, the ensuing covalent bond creates an acetal or kettle linkage between the anomeric carbon of one monosaccharide and the hydroxyl group on another monosaccharide. The final product is referred to as a glycoside. The particular monosaccharides involved and the location of the glycosidic linkage determine the type of glycosidic bond that forms. In sucrose, for example, the glycosidic bond that connects glucose and fructose is formed at position 2 between the anomeric carbon on the glucose molecule and the hydroxyl group on the fructose molecule. A notation like -1,4 or -1,2, which indicates the position and configuration of the anomeric carbon involved in the bond, is frequently used to indicate the specific type of glycosidic bond. As they specify the precise arrangement and orientation of monosaccharide units in a carbohydrate molecule, glycosidic bonds play a significant role in the structure and function of carbohydrates. This, in turn, influences carbohydrate qualities such as solubility, stability, and the ability of enzymes and other biological entities to recognize and act on them. Simple sugars called monosaccharides are unable to hydrolyze further to form smaller sugars. Sucrose, lactose, and maltose are the three most prevalent disaccharides. One glucose and one fructose molecule make up sucrose, and their carbon atoms at positions 1 and 2, respectively, are joined by a glycosidic bond. It is frequently used as a sweetener in foods and beverages and is frequently present in table sugar. One glucose and one galactose molecule make up lactose, and their carbon atoms are joined by a glycosidic bond at positions 1 and 4, respectively. It is frequently utilized as a component in processed meals and is typically present in milk and dairy products. Two glucose molecules are joined together to form maltose via a glycosidic bond at positions 1 and 4, respectively. It is frequently present in grains that are germination, and it's utilized to make beer, make malted drinks, and sweeten processed goods. Disaccharides are crucial for supplying the body with energy because during digestion they are disassembled into their monosaccharide components. Cells utilize the monosaccharides for energy when they are absorbed into the circulation. Disaccharides can also take the following forms. Trehalose is made up of two glucose molecules connected by a glycosidic bond at the 1,1 position. It is frequently present in yeast, some insects, and fungus and is well known for its capacity to shield cells from desiccation. Two glucose molecules connected by a 1-4 glycosidic bond make up cellobiose. It is a part of cellulose, the primary material in plant cell walls. Two glucose molecules connected by an 1-6 glycosidic bond make up isomaltose. It is created when starch is digested and is utilized in food preparation as a sweetener and texturizer. One galactose and one fructose molecule are joined by a 1-4 glycosidic bond to form lactulose. It is a synthetic disaccharide that relieves constipation by acting as a laxative. One glucose and one fructose molecule make up maltulose, which is joined by an 1-4 glycosidic bond. It is a naturally occurring sweetener that can be found in honey and various fruits and is utilized in some meals and beverages because of its low glycemic index. One glucose and one gentiobiose molecule are joined by a 1-6 glycosidic bond to form gentibiose. It is an uncommon disaccharide that is present in some bacteria cell walls. Two glucose molecules connected by a 1-3 glycosidic bond make up laminarabiose. It is a part of the storage carbohydrate known as laminarin, which is a polymer that is present in some algae. One glucose and one galactose molecule are joined by an 1-6 glycosidic bond to form melibiose. It is a substrate for identifying specific bacterial species and is present in legumes like beans. Two glucose molecules connected by a 1-2 glycosidic bond make up saccharose. It is a part of the polysaccharide chitin, which is present in insects and other arthropods' exoskeletons. One glucose and one fructose molecule make up trehalulose, which is joined by an 1-1 glycosidic bond. It is an uncommon disaccharide that can be found in some flowers' nectar and in the waste products of some insects. Disaccharides that perform a number of crucial biological processes that are similar to those carried out by monosaccharides. Disaccharides serve a number of important purposes, including disaccharides like maltose, lactose, and sucrose are vital energy sources for cells and are used as energy storage. They can be broken down by digestive system enzymes to release sugars like glucose and other sugars, which can then be used by cells to produce energy through cellular respiration. Disaccharides also act as support for structure. For examples, cellobiose and laminaribiose, two structural polysaccharides, are crucial building blocks made up of disaccharides. Plant cell walls and other biological structures are structurally supported by these polysaccharides. Some disaccharides, such as trehalose, are involved in osmotic regulation, which helps cells maintain their water balance and protect themselves from external stressors including desiccation. Cell signaling and communication are facilitated by several disaccharides, including lactose and malebiose. For instance, malebiose serves as a substrate for the identification of specific bacterial species, while lactose is an important regulator of gene expression in bacteria. Disaccharides like maltulose and isomaltose are employed in the preparation of food as sweeteners and flavorings. Other sweeteners like sucrose and high fructose corn syrup can be substituted with these disaccharides because they have fewer calories. Disaccharides with qualities that are recognized to be protective and preservative include trehalose and sucrose. While sucrose is frequently used to preserve fruits and other foods, trehalose, for instance, can shield proteins and other biological molecules from denaturation or degradation. Hyaluronic acid, a disaccharide made up of diglycuronic acid and N-acetylglucosamine, provides lubrication and is present in numerous bodily tissues, including the synovial fluid of joints. Hyaluronic acid aids in lubricating joints and reducing bone-to-bone friction. Disaccharides that are important in cell adhesion and cell signaling include chondroitin sulfate and heparan sulfate. These disaccharides are frequently present on the surface of cells and are crucial for cellular communication and tissue growth. Lacto-N-neodytriose and lacto-N-fucopentose are two disaccharides that are important in immune system function. Some bacteria have disaccharides on their surfaces that can interact with immune cell receptors to cause an immune response. Disaccharides, like lactulose, are used as osmotic laxatives to aid in the retention of water in the colon and to soften faces. This can encourage regular bowel movements and relieve constipation. Disaccharides perform a variety of vital and diverse tasks in biological systems, including lubrication, cell adhesion and signaling, immune system activity, and hydration retention.